Hello and welcome to this mirror video to gain-of-function research, loss of function. Loss of function may be something of a misnomer, as functions are not lost so much as the gene is changed to a form which does not work, or works differently. Such alterations can be a powerful tool in developing models of disease, pathologies, and vulnerability. In describing loss of function, we must contrast it with gain of function. We have described how gain of function generally adds a gene or modifies it in such a way as to provide a new function. This covers everything from novel pharmaceutical products to more aggressive pathogens. This is facilitated by either direct manipulation of the genome or by selective breeding under circumstances which contribute to high rates of mutation. In modern research, direct manipulation by CRISPR and other techniques is most common and preferable. Loss of function uses the same methods. The insertion or modification of a given gene or genes. Where gain of function adds to the organism, a loss of function does the opposite. Despite this, it is still described and defined as a gain of function experiment. Any selection process involving alteration of the genotype and the resulting phenotype is considered some sort of gain of function experiment. It's simply a matter of what's being gained. Is it a negative or positive acquisition? One of the most important applications for gain-of-function research is creation of animal models, especially those in the case of pathogens, and how a pathogen works. Notably at the minute, it would be important to understand how something like COVID-19 would enter an animal, and what you could knock out and then prevent from allowing that animal to be infected again. In this case, removal of the ACE2 receptor would do that. It would, however, have long-lasting and wide-reaching side effects that aren't desirable. It does, however, allow you to develop some sort of countermeasure. In fact, having an animal that is vulnerable to a disease is essential if you're going to try and get whatever it is you're creating approved by most, if not all, drug approval agencies, such as the Food and Drug Administration. Most of them have some sort of requirement that's going to follow the general rule of having the disease that you're trying to treat infect the animal, and that the animal model mimics the human in terms of what gene it has or doesn't have. Taking the premise of removing a gene's functionality, this is known as a knockout experiment. And it's known as that because you remove the activity of the gene. This gives incredibly accurate and important information about what the gene would normally do, and in some cases how it interacts with either other genes or other cellular processes. We're fortunate, at least in research, that the overlap between genes in a mouse and a human are considerable. A simple Venn diagram, although not exactly ideal as a description, would largely overlap, with a few notable exceptions. By taking a mouse and conducting knockout experiments, you can begin to understand just how a gene, whether it's the same gene as in the human or the mouse variant of that gene, you can figure out what it contributes to disease and other pathologies. This is important to remember. But there's a big caveat to gain-of-function research that uses a gene knockout. Approximately 15% of all the known genes that are functional will be required for life. That is, without them, the mutation is lethal, and in fact developmentally lethal. That means the embryo cannot turn into an adult, no matter what you try. This can make it very difficult in some cases to work on developmental disorders in an animal model. Further to that, if we're talking about either an acquired or very slow-acting developmental disorder, the animal may not have the opportunity to grow into a mature form, and therefore we're unable to observe the effect that that particular mutation has in the adult. This is important if you're looking at diseases that progress over someone's lifetime, where they eventually get worse. What impact does it have on a developing child and a mature adult? Then we need to look at what changes may happen under normal circumstances. Removing a gene may not cause any harm in all cases. Notable examples of this are those involved in cancer research. Cancer is a statistical certainty to a certain degree. Most people will have some interaction with it in their life, and the fraction who don't are both incredibly lucky and unfortunately ignorant. If you were to modify just one cancer-related gene, like P53, you might find that the number of cancers in the mouse population soar, but you may find that there are some who simply demonstrate no noticeable change, and this is all down to a numbers game. There are also issues around just what sort of cancers will develop. As we've mentioned, just because you have the same gene in a mouse and a human, the gene may not necessarily function the same way. P53 is an example of this, where the tumours will develop in a different range of tissues in the mouse versus the human. Even with that in mind, by various counteracting measures, such as humanizing the mice, you are able to counteract most of the effects, and the benefits of a knockout experiment clearly outweigh any downside from it. This is all the more true if you're going to be looking at new pharmaceutical interventions that are intended to prevent that. It also provides some insight into the flaws of animal models, and where future research will need to be directed to improve the representation of the human in the mouse. There's also something known as a conditional gene knockout. This is a very interesting way of going about things, and that's because it's clued into a timer. That is, the knockout will only occur if particular criteria are met. This is important when you have those mutations that are essential for embryonic development. If you want to develop an organism from an embryo to a mature adult, and then find out what deactivating that gene will do, you simply can't have a genetically modified cell line or animal line, as they would never be able to grow to maturity. Since you can't have this working in the embryo, you need to be able to activate the nullification of that gene at some other time. One of the more common methods for this is the LOXP. These are short sequences around the gene that you want to remove. By having two different germlines, one with the CRE recombinase, which is a viral enzyme that recognizes that particular structure, the LOXP, and the other carrying the gene of interest. You can, in theory, have the viral enzyme activated later on, and that then removes the particular gene in question. That allows you to decide when and how the gene is deactivated. Recent examples of this in practice include the modification of mosquitoes to be sterile if male, and in other cases, to prevent female reproduction or feeding. Mosquitoes are known to spread diseases across the world. Dengue fever, yellow fever, malaria, there's a long list. Just one example of this is a gene known as double sex. This is essential to the sexual development of the mosquito. It leads to the female mosquito being more masculine. You can observe these effects in the long protuberance that allows them to bite you and feed on your blood, growing shorter and shorter. Their mouths increasingly resemble that of a male mosquito. Since they can't bite, they can't do several things. One is they can't feed. Second, importantly for humans, is that they can't spread disease because they're not moving between humans and injecting whatever it is they're carrying disease-wise into a new host. When you account for these factors, you've already greatly diminished the female mosquito's ability to carry disease. But then the reproductive organs themselves are damaged by this. That means that they don't reproduce readily, and in some cases cannot lay eggs at all. As more and more mosquitoes are born, more of the female population has this modification, and as a result, an ever wider portion of that mosquito population becomes sterile. We then have the recreation of a propensity towards cardiovascular disease, notably atherosclerotic plaques, in animal models such as rats and mice. Generally, mice are very resistant to develop of plaques and lesions, and this would mean that they were very inappropriate to use as the basis for a study. There are a couple of ways this can be fixed very quickly and very effectively, and why they're favoured as a research animal. It's a stable mutation. By modifying lipoprotein carriers and parts of the metabolism pathway, along with feeding them a high-fat diet, you're able to make them prone towards hypertension and atherosclerosis. The low-density lipoprotein receptor deficient mice, or LDR negative mice, and the apolipoprotein E deficient mice, or ApoE negative mice, are commonly used because of that stability. You can further exacerbate the development of these plaques by initiating diabetes or hypertension, if it's not already being caused by the development of these plaques. Finally, there are the developmental models we've already spoken on for a short time. You take a gene essential for growth and let it work as planned until a specific time or under specific conditions. Pitt-Hopkins syndrome is an example of this in humans. It involves a mutation in the TCF4 gene. The gene provides specific instructions for creating proteins that are needed for many functions of the body. It leads to notable changes in the face, development, and neurology of people who have this. And then we have the ever-entertaining Kenny gene. The Kenny gene, or oh my god, they killed Kenny, was named by a Strasbourg scientist and South Park fan called Sophie Rushman. It's commonly found in fruit flies, and what happens is those fruit flies that inherit this gene will die within 2 days of being infected by a bacterium. This is one of those incredibly useful genes where, if you're trying to figure out what roles are played and how development occurs and you want to stop it suddenly without any other notable changes, you can introduce the bacterium and it initiates the death of the fruit fly. It also allows you to study the involvement of certain genes in development, where they play a role, what they do. The Kenny gene in particular is involved in the immune response, and so when you look at the Kenny gene in conjunction with another, you can see that there's an interplay of different parts of the genome in defense of the fruit fly body. Knowing that the Kenny gene is absolutely essential for activation and working of the immune system allows you to then look at what cascade occurs, what effects it has, and why it's doing what it's doing. In summary, gain-of-function research when used to lose functions is not something bad, but it is a matter of application and purpose. As we've already stated, in some cases, research can be used for nefarious purposes no matter who you ask or why it's done. There's simply no oversight in some cases. In other instances, it's necessary to have an organism lose certain protections so that you can investigate how a particular pathogen works, what it does to gain access to a cell. If removing that particular gene means that pathogen can no longer gain access to that cell or organism, does it remove the vulnerability, and how can you recreate it without having to genetically modify the organism? Gain-of-function is not just limited to creating what is imagined bioweapons and other conspiracy theories. Rather, it exists in a much larger framework. Thank you for watching this video. If you found it interesting, consider liking, sharing, and subscribing. Please post any comments, questions, or suggestions that you have below.